Thank you, Dr. Harkrishnan. Am I audible?
Yeah, you are audible. Please go ahead. Can you share your
First of
screen?
all, First of all, thank you very much, Dr. Harkrishnan, Dr. Karta, and Dr. Jay Kumar for organizing this wonderful uh, meeting. And thanks a lot for making me a part of it. So now I'm sharing my screen. So is it visible now?
So we have actually
It is
lost
visible.
your, we
Please
have
go
got the
ahead.
presentation,
Yeah,
but we have
thanks
lost
a lot.
your image, sir.
Yeah, now I will speak
Start your
about non-coding transcriptomics of heart failure. So here I will take an example of one non-coding RNA which we identified as regulated by a cardiac transcription factor, NPAT. This NPAT has been shown to be important for many cardiac disease pathological conditions, including heart failure. So when you look at the human genome, we know that majority of the genome is transcribed, and yet very little portion of the this transcriptome is translated into proteins, and majority of the transcribed RNA remains as non-coding RNA. And previously, it was believed that this non-coding RNA is kind of a junk and it doesn't have any physiological significance. It is just as transcriptional noise. Now we know that this non-coding RNA plays very important role in many physiological and pathological processes, and we are focusing on role of or importance of the non-coding RNAs in cardiovascular diseases. Especially, we focus on long non-coding RNAs. So when you look at the key features of heart failure, so there are many features. I would say one of them is the re-expression of fetal genes. The genes that were embryonically expressed, they are kind of expressed again during heart failure. And most important feature is impaired calcium handling. So this is mostly because of downregulation of one particular gene called sarcoplasmic endoplasmic reticulum calcium ATPase. So it is a calcium ATPase that is located on sarcoplasmic reticulum. So if you look at the healthy and failing heart, in healthy heart you have abundant SERCA expression and calcium that is released from the ER lumen to cytosol. This is intracellular space. Is quickly taken up by the SERCA again into the ER lumen, and this cycle goes on. So calcium comes out from LTCC, L-type calcium channel, and it is taken inside by SERCA. In failing hearts, you have severe downregulation of this ATPase, SERCA, and the calcium that is released from ER to lumen, it remains in the cytosol, and this calcium then activates many of the pathological signaling processes, including activation of NPAT signaling. So when you look at hypertrophy, we have physiological hypertrophy and pathological hypertrophy. So physiological hypertrophy is characterized by increased angiogenesis and it is reversible, and we also have increased ATP production, whereas pathological hypertrophy has impaired calcium handling and mitochondria of this mitochondria in these hypertrophic conditions are dysfunctional. So this is how the lumen of the heart looks like in pathological hypertrophy. So you have dilation here. So here I will talk about one of the work of one of my graduate students, Priyanka Pan. So she is investigating the role of certain NPAT-regulated link RNA in hypertrophy. So as I said, SERCA is one of the genes that is frequently downregulated in hypertrophy, and both there is preclinical and clinical evidence that SERCA gene therapy is beneficial in hypertrophic conditions. So in fact, there are some clinical trials still going on with this gene therapy model. And here we took three groups of rats, whereas one is control and one is rat with failing hearts, and one is failing heart rats which received SERCA2A gene therapy. And we did transcriptome profiling for link RNAs. And you see there are several link RNAs that are upregulated and there are some that are downregulated. These are link RNA candidates. And yeah, you see there is one link RNA here which I represented with a red dot. So this is comparison versus control versus failing hearts. And similar comparison we did with the control versus failing hearts that received SERCA2A gene therapy. Many of the link RNAs that were deregulated in failing hearts, some of them could be restored back to the normal levels when we gave SERCA2A gene therapy. You can see this red dot comes here. And presently we are calling this link RNA as L72 because that is our internal primer number. And we also verified this induction of this L72 link RNA with individual qPCRs also. You can see that this link RNA is upregulated during failure, and when the heart function is restored, the link RNA comes back to normalcy. And we also have data to show that heart function is completely restored after circuit therapy. So we also did some in vitro assays. So we used here one agent called toxicogene. It is a pharmacological inhibitor of SERCA function. So we took H9C2 cardiomyoblasts, rat. And when you treat cells with different concentration of this toxicogene, you can see that a gradual buildup of this link RNA. And when you inhibit this calcium upregulation by toxicogene using a calcium chelator, BAPTA, you can again set back this induced link RNA to normal level. So you see that this L72 is induced upon toxicogene treatment. This is BAPTA alone, and when you treat toxicogene plus BAPTA, this link RNA comes down to the near to normal level. And this is a positive control which we use for activation of, inhibition of SERCA by toxicogene. Whenever there is SERCA inhibition, you see this stop expression going up. And also we treated cells with isoproterenol and phenylephrine. You can see that cardiomyocytes is increased after this treatment, which is denoted here. And also upon these conditions, increasing concentration of isoproterenol increases expression of cardiac stress marker, which is ANP, and which is also parallel by increased expression of this L72. And we did extensive cell culture work to show that in different cell line, this is rat cell line, H9C2, AC16 human, HL1 or rat, mice, sorry, and also in primary rat cells, using different stimulants, isoproterenol plus PE, and also norepinephrine. We see constantly increased expression of cardiac stress marker, ANP, and also always L72 is always upregulated in all these settings. So it also gives us an idea that L72 is somehow related to cardiac stress. And we also used in vivo models. One of the models is isoproterenol injection. So we used the different concentrations of isoproterenol, 15, 30, and 50. You can see that heart is enlarged after 10 days of injection, cardiac hypertrophy. And at all these conditions, you also see upregulation of cardiac stress marker, ANP. And you also see gradual buildup of L72 over time, concentration dependent manner. And again, we also used TAC model, transverse aortic constriction model. So where we placed a hemoclip here. So this is aorta, and you can also see that the clip is placed here, the small metal shiny thing here, and also Doppler confirms that placing of the clip and the blood flow is affected, and you can see that there is already dilation of the ventricle. And under these conditions, this is also a cross section of the heart showing that there is indeed increase in cardiomyocyte size, and this is the heart, total heart. And we also have increased expression of ANP and BNP cardiac stress markers at all the time points, 4, 8, and 12 weeks. And according to that, we also see that after 8 weeks or so, the L72 also started to build up in the hearts. So now we have in vitro data showing that L72 is expressed in response to cardiac stress in cultured cells, and also at least 2 models of in vivo models of cardiac hypertrophy. And when we look at how is L72 regulated, so we see at least different peaks for NPAT-C3 binding. And what we suspect is upon increased cytosolic concentration, cytosolic calcium concentrations, calcineurin gets activated, which activates NPAT, finally leading to pathological programming of the cardiomyocyte. So we to test this, we treated cells with ionomycin because this is also an agent which induces intracellular buildup of calcium. And you can see that RCAN 1.4, this is classical example of NPAT-regulated link RNA. So it is severely induced upon ionomycin treatment. Similarly, you also see L72 upregulation under these conditions. And also similarly, you also see RCAN 1.4, increased expression of RCAN 1.4 upon isoproterenol treatment. which also shows that under these conditions, NFAT is active because NFAT is the transcription factor that drives transcription for RCAN 1.4. And you also see buildup of L72 here. And this is cardiac stress marker ANP. And we also took one constitutively active NFAT. This is NFAT, and it has one serine-rich region which is phosphorylated by several kinases. So upon phosphorylation, it is always active. Sorry, the phosphorylation causes NFAT to be inactive. So there is one constitutively active NFAT vector where this particular SRR region is deleted, so that the NFAT is always active. So we, as I said, we observed at least 2 binding sites for NFAT in the promoter of L72. One is at around 1.8 and one is at around 4 kb. So we constructed both these luciferase vectors with these promoter regions. As you can see that when you express this constitutively active NFAT, the promoter luciferase activity goes up both at 1.8 and 4.1, saying that this promoter is responding to NFAT activation. And also we see that silencing of L72 represses some of the cardiac stress markers, at least A and B. So this is H9C2 cells where we silence the L72 using siRNA. So you see that upon L72 silencing, ANP expression drastically reduces. And we also created one CRISPR-based knockout cell line, AC16. Here also you see that when you silence, when you completely genetically remove this L72 from the genomic loci, you will also see that downregulation of ANP even upon isoprotein treatment. So what is the mechanism of action? So we, for that we did RNA sequencing from this CRISPR cell line. So you see that this is the chromosome where L72 is transcribed. So you have both upregulated genes and downregulated genes. Interestingly, the region where it is transcribed, all the neighboring genes are downregulated. So especially for this region, if you look closely, this is how it is. So almost all the neighboring genes, there are around 180 or so, all of them are downregulated. So there is hardly any upregulated gene in this region. So when you move away from this transcription start site, you see both up and down regulated genes, but around this region, there are particularly down regulated genes. And mostly this region is enriched with many genes that are important for cardiomyocyte functioning. So one of those interesting genes is GRK2. This is G protein coupled receptor kinase 2. This is nothing but beta adrenergic receptor kinase 2, which actually regulates beta adrenergic receptors. We all know how important are beta adrenergic receptors in cardiac pathology. And inhibition of GRK2 has been already proposed as a candidate for heart failure. There is extensive literature arguing that are debating that GRK2 is a potential candidate for development of novel therapeutics for heart failure. And there is also one clinical trial that is ongoing now, which is aimed at inhibiting GRK2 for benefit of for improving heart function in myocardial infarction. So what we found is under these conditions, we also see upon using independent experiments also, silencing of L72 results also in silencing of GRK2. So what we believe is that at this moment, the experiments are still ongoing. We think that this L72 recruits some coactivators to its neighboring genes, thereby enhancing the transcription. And in the absence of the L72, these coactivators are not being recruited, especially P300. We also see signatures of P300 binding around this GRK2 region. So we believe that this L72 works by recruiting cofactors to the neighboring genes. And in the absence of this L72, these cofactors are not able to bind to this GRK2. That's why the expression of these markers is going down. So we are presently doing experiments in that direction, and we will also soon do knockout animal study with L72. So we will soon start experiments in those animals. And in summary, this link RNA L72 is upregulated in different models of cardiac hypertrophy and failure. And the cardiac pathological transcription factor NFAT drives the transcription of this L72 in response to, mostly in response to calcium handling in cardiomyocytes. And L72 may function by regulating beta-arrhythmic receptor kinase, that is GRK2. Thank you. These are my present lab members, Abhishek, Disha, Priyanka, Sindhura, and Tripti. And Priyanka is the one that is contributing to this work here. So this is her graduate project. And I acknowledge funding received from DBT Welcome. And I still collaborate with my previous institute, Hanover Medical School. Thank you.
Thank you, Dr. Kumaraswamy. Dr. Karta, please.
Regalla. Thank you, Regalla, for a very interesting presentation. This Regalla's presentation actually gives us a feel of what RNA biology can do to find new avenues for treatment of cardiac failure. I think we will move to the next speaker because already the time is up.
Okay. Yeah, thanks, Dr. Karta.